Okay, excellent. Thank you very much, Olivier, for the invitation. I hope you can hear me sound clearly. Otherwise, stop me, interrupt me.
Yes, we can hear you very well. We can hear you very well.
Excellent. So yeah, I'll tell you briefly about myself. Currently, I'm a senior scientist at the Institute of Human Nutrition and Food Science, the University of Kiel in Germany, north of Hamburg. I don't know how many of you are familiar with that, but so I have been associated now full-time with the institute since the beginning of 2016. Before that, I worked for almost 11 years for the industry. And as a researcher in the innovation department of different companies. I work in Europe for about 8 years, and before that I work in the US for Cargill, always in research. And originally I'm from Argentina. That's where I grew up, and then I got my Master of Science and PhD from the University of Illinois in the US in nutritional physiology. I work in ruminants, in dairy cattle mostly. So since you know, when I started working for the industry, I moved into all kind of species, not only ruminants. That's where my scope, you know, my interest broadened. Since then, you know, most of my time I have been deeply interested in gastrointestinal physiology. And in particular in intestinal physiology. And some years ago, we started some collaborations on research that led us into the topic of today. And the slides that I have presented here will help me define and tell you a little bit more about how we got interested in this topic and how we learn about it. So it's okay if I go on, Olivier, is that right?
Yes.
Excellent. So okay, just to set the context, I'm going to tell you 2 or 3 things about, you know, how what I'm going to be talking about, what do we understand for antimicrobials, and in which context we are going to be talking about them. So for us, within this presentation, when I use the word antimicrobial, I'm referring to antibiotics and also to heavy metals like zinc oxide, copper sulfate. Mostly, they have been used for many years to promote growth, to stimulate growth, in particular feed conversion in pigs and in poultry mainly. But also they have been used for disease prevention, particularly enteric disorders that are associated in pigs with weaning, for instance. The way they have been used, it's important to keep in mind that antibiotics always have been used as, and the use I'm referring to is the subtherapeutic use, that is doses below the therapeutic level needed for treating a disease. And in the case of the heavy metal is the other side of the equation. They have been used as supranutritional levels, so many times higher concentrations than what are actually needed from a nutritional standpoint. The way of delivering them has been mostly through feed, but sometimes also through water, particularly in poultry operations. And they have been used, like I say, for many years. The use actually skyrocketed in the mid-late 40s, and that was coinciding with the release of new antibiotics to the market. So since then, it has been heavily used to you know, there are some estimations based on data from 2013 These estimations indicate that the consumption globally of antibiotics, estimated for 2017 was 131 tons globally. 50 to 80% of that is projected to be consumed by animal production. 75% of all consumption is happening in 3 countries where actually most of the animals are being produced, China, USA, and Brazil. But as you all know, this is history, this is changing. We are all in a quite different mindset. Worldwide animal production is, right now is engaged in demedicating production. Main driver for this is, of course, antibiotic resistance or antimicrobial resistance, which if we don't do anything, projections are telling that probably there are going to be about, in 2050, there's going to be about 10 million deaths from antimicrobial resistance, which are going to become the number one reason of death, human death, surpassing things like cancer, diabetes, and others. So there's a lot of interest, and again, this is simply set in the context. I want to add something here also in the interest that is growing over the last few years. It's a more recent thing. There's a lot of epidemiological data showing that exposure of to antibiotics early in life, I'm talking about humans, it's also associated with weight gain and obesity, similar like we have been documented in animal production, but also is associated with long-term issues related to health, like allergy, autoimmune disease, and also susceptibility to infectious disease. So this topic is becoming interesting not only for animal production or people or scientists related to animal production, but also for people interested in human medicine and health. So when we became interested in this topic, essentially I was working for the industry. The driver was, along with some colleagues from the academia that I interacted, we were saying, okay, we need to find alternatives, and which could be a good opportunity for the industry. So we did this, we went to review the literature, and we say, okay, what do we know about it? The number one thing that came up to our mind was that response is very consistent. It doesn't matter the bacterial spectrum, the diet, the rearing conditions, the type of animals, antibiotics, subtherapy, or antimicrobials, they seem to deliver consistently that response. A response that, depending on the animal, typically ranges from 9 to 10, or let's say from 5 to 10% performance improvement, that is growth or feed conversion. So this suggests that there has to be a converging point. When we discussed the mode of action, we were saying, okay, there has to be a point where all these compounds, when they are offered or included in the diet, they trigger this consistent response. And we know that with the animals that we work, if something is not consistent. So where is the converging point? So if you go to the literature, you're going to find that the most frequently effects that have been reported in the literature are, number one, altered gut microbial ecology. Some people have demonstrated that antimicrobials reduce microbial load, sometimes pathogens or opportunistic bacteria, but they also reduce microbial diversity, because most of the antibiotics and zinc oxide and copper sulfate, they are known to target gram-positive bacteria, and they favor the proliferation of high energy yielding communities. So that is an interesting and a very logical and plausible effect. However, this effect certainly is not universal. if you go around and you look at the literature, you're going to see that there are reports showing that you can have the phenotype, you can have the growth promotion without changes in gut microbial composition. Sometimes there's some people reporting improved growth with increased gram-positive bacteria, opposite to 90% of the other research. And therefore, there doesn't seem to be or exist a growth-promoting microbial fingerprint. So I'm discussing taxonomy here. And so that is not a converging point. At least data doesn't suggest that actually a microbial fingerprint or enterotype is actually the converging point. This led support to some theories. One of them is another alternative is that probably the growth promotion comes from the the anti-inflammatory effect of antimicrobials. And this theory, has some support. There are some data supporting this idea, and the anti-inflammatory effect seems to be one indirect, but also a direct effect of antibiotics directly acting upon immune cells to reduce the inflammatory response. The result of that lower degree of inflammation is nutrient sparing for growth. So that's the whole idea. However, I believe that in 2014, a very well done study showed that antimicrobial growth promotion is actually linked to gut microbiota in a cause-consequence relationship. So this study by Cox and co-workers showed that what they use is they treated mice with low doses of penicillin, and then they transfer the microbiota from the antibiotic-treated mice to germ-free mice, and they were able to show the growth promotion, and that the growth promotion was associated with increased adiposity. This study, when you look at the mechanism that could be related to that, they show reduced intestinal inflammation, so lending some support to the anti-inflammatory hypothesis. But they also show very interesting, this was another study from the same group. They show increasing adiposity and a very large alteration of the lipid metabolism. They look at different genes and pathways involved in lipid metabolism, and they were able to show that. So that triggered the question: how is that altered gut microbiota is actually inducing these effects? And by the mid-2000s, information was accumulating, indicating a link between gut microbial dysmetabolism and inflammation. And so today we know that there are some estimates that about 10% of the circulating metabolome comes from the gut microbiota, but we don't know what is that. We just know few of the metabolites produced by gut microbiota, like BFAs or vitamins or derivatives of indole, but many of them remain unknown. And there are three sources of essentially metabolites. One of them are the dietary components that the animals ingest and the microbiota transform. The other one are host metabolites that the gut microbiota transform, and by the other group belongs to the ones that the gut microbiota produce from de novo. So with all this in mind, we developed a working hypothesis. We propose that antimicrobials actually they target the communication, the dialogue between gut microbiota and host, and we say that they target the content. And what I mean by the content is the signaling molecules that are exchanged between gut microbiota and the host. And we propose that that could be a converging point. That could be where antimicrobials, it doesn't matter the antimicrobial spectrum or the situation, they converge. This effect happens not only locally at the level of the intestine, but also has far-reaching consequences, distally targeting the liver, the muscle, the fat. And the consequence of this change in the content of the crosstalk is likely a resetting of the immunometabolic processes that are associated with growth. When you think what molecules, could be responsible for that, we thought that bile acids were a very nice candidate for that. So I'm going to tell you a little bit about bile acids before jumping into our research. And so briefly, bile acids are produced in the liver from cholesterol. There are two biosynthetic pathways. One is called the classical, you have it in the left side, mediated or initiated by CYP7A1, which is the sort of the flow control, the bottleneck of this pathway. And this pathway is the one that produces generally the largest amount of bile acids in health or in steady state conditions. We also have the alternative pathway, but that is less relevant. The bile acids that are produced by the liver belong to two, let's say, chemical structures. One of them are dehydroxylated and called quinodeoxycholic acid, the one on the right. And then downstream of CYP7A1, bile acids can be hydroxylated in position 12 to become cholic acid. So these are the two main primary bile acids. We call primary those that are produced by the liver. However, when bile acids, enter in the gut, they become exposed to gut microbiota. It is not really known why, but there are different theories about why gut microbiota has such a big capacity for transforming them. So the quantitatively speaking, the two largest modifications that we know are the one that you have there highlighted mediated by the bisalt hydrolase, which is the deamidation, so releasing the taurine or glycine from the bile acid. And then the second most common or prevalent microbial metabolic transformation of bile acids is called 7-alpha dehydroxylation. So that is removal of the hydroxy group in the 7-alpha position of the bile acid. So when you do that, dehydroxylase CDCA becomes lithiocolic acid, and when you dehydroxylase cholic acid, it becomes deoxycholic acid. So these are the four most common bile acids in a number of species. But it's important to keep in mind that the bile acid composition, so the chemical structure, shows a lot of variability, and there are a high degree of specificity among species. So the bile acid profile of pigs differs significantly from that of humans, and that one significantly from the one of mice, and so on and so forth. As soon as bile acids are synthesized in the liver, they are stored in the gallbladder during fasting. So whenever a meal is initiated, cholecystokinin, that is released mostly from the stomach, induces the contraction of the gallbladder, which dampens the bile content into the duodenum. As bile acids travel through the intestine, they are able to participate in the digestion and absorption of lipids and fat-soluble vitamins, and eventually most of them are absorbed as salts, mostly from the terminal ileum. Some of them, that's about 95% of them are absorbed as salts from the ileum. The 5% remaining, let's say, escapes and enters into the large bowel, where it becomes, subjected to microbial modification. So the dehydroxylation I mentioned before, for instance. And in that case, then they can enter into enterocyte through passive diffusion. So bile salts and bile acids, all of them enter eventually into portal circulation. They go back to the liver. The liver takes them up very efficiently, but right after a meal, sometimes there are some leakage, some spillover into systemic circulation, but eventually those bile acids return to the livers through arterial circulation, blood. So normally this data you have here on the right, bottom right corner for fecal excretion, it's for humans. Very low amounts are lost, and that amount is made up by biosynthesis, de novo biosynthesis by the liver. So the bottom line is that the enterohepatic circulation that I just described is a very efficient process, very well at conserving bile acids, which remain a very constant concentration throughout the day. Well, our knowledge about bile acids experienced a huge leap forward since the late 90s when bile acid devoted receptors were discovered. The first one was farnesoid X receptor, which is a nuclear receptor. After that, others nuclear receptors were added to the list of bile acid sensing, and also some G protein couple receptors. The number one devoted to bile acids is called TGR5 or also GPR1. But the interesting thing here to keep in mind is that these receptors are widely expressed within the enteropathic system, but also outside the enteropathic system, like in adipose tissue, like in brain, like in adrenal glands, so very, very extensively expressed. They have different sensitivity to bile acids. FXR is mostly sensitive to primary bile acids. As you can see, CDCA is more potent than DCA, which is a secondary bile acid. However, TGR5, which is a G protein couple receptor, is more sensitive or more potently activated by secondary bile acids, so LCA, DCA, so on and so forth. Well, what we have learned through the last 10 years is that by acting upon these receptors, bile acids are known now to integrate the responses to nutrient intake. They modulate the immune response and the inflammatory response immediately after a meal is taken. They do that by, on one side, avoiding bacteria overgrowth and risk of dysbiosis right after a meal. But they also act upon mechanisms that enhance intestinal protection against microbiota, pathogens, toxins, and other antigens. They enhance barrier function, and they also have anti-inflammatory effects by acting upon receptors mostly expressed in immune cells present in different parts of the body. Bile acids also have been shown to modulate metabolism. They are known to, by activating FXR and TGR5, but also working with FGF19. I'm going to explain that in more detail in a little bit. They shut down hepatic lipogenesis and gluconeogenesis right after a meal. They increase glycogen and protein synthesis, insulin secretion and sensitivity, and they stimulate lipid storage and energy expenditure. All that within the feeding fast cycle. Well, the first group that I believe became interested in the possible role of bile acids in animals was a group of Doug Behring in the Baylor College of Medicine. They pretty sure some of you know him. What they work with premature piglets, 2 days old, that they are fed via total parenteral nutrition. Under those situations, the gut suffers atrophy, as you can see it on some of the pictures there. And what they show is that by including CDCA in infusion, so infusing CDCA in the intestine of these animals while they were receiving total parenteral nutrition, sort of rescue the intestinal structures, so stimulated intestinal growth, so also stimulated, in particular, the growth of the intestinal mucosa, and that was associated with a recovery of the secretion of the circulating levels of glucagon like peptide 2, GLP-2, which is the intestinotrophic peptide. So because GLP-2 has been implicated also in inducing some of the growth check and the enteric disorders that are associated with weaning in pigs, we became interested in this, and so we decided to run our very first study, actually in collaboration with Doug's team. So early in 2012, we ran this experiment where we took weaned piglets, 3-week-old, normally managed and fed, and we simply infused them with CDCA, hoping to see some positive effects. So we were able to show the GLP-2 response, as you have it there on the left bottom corner. We were able to show GLP-2 response, but that was not really associated with any improvement in animal performance or in gut health or in gut maturation and function, except for a few trends. So we were sort of disappointed, probably because we did not do things right at that time. But right at about the same time, more and more evidence was mostly from the biomedical field was accumulating, indicating that two very important things. One, that antimicrobials affect the synthesis and tissue distribution of bile acids, and two, that such alterations are associated with alteration or changes in the metabolic and immune homeostasis of the host. So with those two things in mind, we rethought our ideas and reconsider what we have learned, and we decided to go ahead and initiate a new line of research oriented to learn more about bile acids in farm animals. We started working with piglets as our model. We took normal pigs, from commercial operations. They were weaned on average at 21, 3 weeks of age. So typical management, nothing special here to highlight. Diets that we used were very simple, cereal-based, no medication use in the control diets. And then we decided to use as antimicrobial, model. Let's say we decided to use combinations, and the reason is because up to that time, when we started or when we conducted these studies, in most part of the world, I would say, antimicrobials were not using as a single intervention. They were using mostly a combination of either two antibiotics, sometimes antibiotics plus zinc oxide. So we decided to use a combo that was very popular, let's say, in the south part of Europe, which is the combination of zinc oxide with amoxicillin or colistin. That was study number 1. And in study number 2, to sort of validate and expand our findings, we decided to use zinc oxide in combination with chlortetracycline and thiamulin, all at doses that are considered, subtherapeutic. And all this work has been published, and you have there the citation. So the first thing that we wanted to be sure that we were getting was the phenotype. And so we look at growth, and we seen the response that was expected. In both study, body weight improve over time, reaching at the end of the transition period, which was the end of our studies, reaching about 10, 9, 11% more body weight. That response was not associated with changes in feed intake, which are the two graphics on top, meaning that feed conversion was improved. So prototypical consistent effect of antimicrobials, as has been reported and as people is used to observe in the field. So the first thing we did was, of course, to measure gut microbiota, to see if we were able to find the enterotype that we wanted to see. So in the study 1, which is reported on the left side, the ZAC intervention gave us exactly what we were looking for. So reduce diversity, reduce gram-positives, and so reduce lactobacilli, for instance. They were knocked down, some Clostridium as well. So that fitted very, very well with what we were expecting to see, and some people have described. However, when we analyze the second study, which is on the right side of the slide, the ZSCT intervention did not show any of that. So no changes in diversity, some numerical changes in the same direction for gram-positive bacteria, but nothing really significant, and certainly not close to what we observed there. So it was adding confusion, but was in line with what I mentioned about the literature. So the next step was to look at the bile acid world. So we started in the gut. We look first at the bile salt hydrolase activity, and the reason is because most of this activity, the BSH activity, it is granted by gram-positive bacteria. It is a universal type of activity, but it's very prominent in lactobacillus, for instance. So if antimicrobials knock down lactobacillus, you expect to see also a depression on bile salt hydrolase activity, which has been described in the literature and has been proposed to be part of the reason why antimicrobials improve growth. So on the left, again, you have the first study. We found that depression on bile salt hydrolase activity, happy again, but when we look to the right side, we found the opposite. So no convergence, no reasonable explanation so far. So we continue on and we start analyzing bile acids at different, in this case at the colon, so in colon contents and tissues. And this is the first eureka moment to us, because what we found was that both interventions resulted in very similar changes in 7-alpha dehydroxylation activity, which is here represented by a clear increase in the dehydroxylated 7-alpha dehydroxylated bile acids, which are HDCA, which stands for hyodeoxycholic acid, and LCA, which is the lithocholic acid. So this was for the first time in our research, comparing these studies, which were conducted 2 years apart, I should say. For the first time, we were finding some consistency. Then we decided to go on and say, do these effects actually expand into other tissues? And so we look at several tissues. Here I'm reporting liver, plasma, ileum, colon. So these are tissues, and in tissues we were observing some consistency as well, that the oxycholic acid was increased in liver and in the intestine, and that lithocholic acid, the LCA, was increasing mostly everywhere. the importance of this finding, on top of being, the same for each study, that the importance is the fact that these bile acids are the most potent agonist of bile acid sensors. Like I explained before, CDCA is the most potent activator of FXR, and LCA is the most potent activator of TGR5, telling us, or this is cluing or giving or suggesting that, we should expect enhanced bile acid signaling. So with that in mind, we went on looking for targets, of bile acid sensors that have been described in the literature that are typically regulated by bile acid sensors. So we find that these bile acid sensors were present and affected in the direction that were expected. We found reduced production of inflammation mediators, also like interleukin 8 or COX-2, which are prototypical targets of FXR and TGR5. We also found increased expression of some antimicrobial peptides, which is ANG1, which is a target of FXR. And on top of this, we look at the physiology of the intestine. We look at ileal permeability and the ion transport capacity using chamber studies, and we found that actually bile acids were working, that the antimicrobials, which included zinc oxide, were working in the direction that we were expecting, so reduce secretory capacity of the intestine and improve barrier function. So, we went on, like I say, that we were able to find that the effects of, or the changes in bile acid profile were affecting other tissues. So we decided to look into different tissues. We look at subcutaneous and visceral white adipose tissue, and we also found signals, let's say, of reduced inflammation in adipose tissue. It's important to, which is, this is indicated in the left plot. It's important to mention that TGR5 has been implicated in enhancing thermogenesis, so in adipose tissue. So we look for signs of that, and we did not find any. So there you have different target, proteins that are affected by TGR5. None of them were affected. So this data suggests that antimicrobials, as used in the context of promoting growth in animals, are not associated with enhanced energy expenditure in adipose tissue, but yes, reduce inflammation. So up to this point, our data was giving support to the antimicrobial, anti-inflammatory hypothesis of growth promotion by antimicrobials. So basically, that reduced inflammation may spare energy and other nutrients for promoting growth. But some data particularly from Kirk Lansing in California is indicating that some of the cause, or the changes associated with inflammation are not really associated with changes in systemic immunity, but rather with inefficiencies at the metabolic level. So that makes us think that probably we should go on and looking for a connection between metabolism and or changes in metabolism and the changes in bile acid that I just mentioned. So we look at the liver for bile acid signaling, and we found what we expected. That's on the left, the genes that you are seeing there, the change in expressions, the SHIP, SHP, and the SRBP1C. These are target genes of FXR. meaning that FXR was activated. And on top of that, we measured, the composition of the liver, and we found decreased accumulation of triglycerides, or reduced lipid accumulation in liver, but increased protein accretion in liver. This is suggestive that not only bile acids were acting upon FXR, but also through FGF19. FGF19 is called fibroblast growth factor 19. It is produced in the mostly, I should say, in the terminal ileum in response to FXR activation. So we look at that, and as you can see on the left side, we found increased expression of FGF19 protein and also gene. It's not reported here, but we did find it. And we did find increased FGF19 in circulation in both data sets, in both studies. FGF19 is an enterokine that signals through receptor, which is an heterodimer between FGFR4 and beta-klotho. We measured those receptors in different tissues. Here I'm showing data for skeletal muscle and white adipose tissue. As you can see, they were not affected by FGF19 or by the antimicrobial feeding in this study. So suggesting that it is possible that some of the effects that we were seeing may be these endocrine effects mediated via activation and release of FGF19. Why is that? Well, FGF19, simply put, is an insulin-like hormone. It's involved in shutting down or inhibiting bile acid synthesis, but it also has metabolic regulation effects. The difference between FGF19 and insulin is that FGF19 does not have the lipogenic effect of insulin. That is because they signal through different pathways, insulin through AKT, mTOR, whereas FGF19 uses a different kinase pathway, which is not linked to activation of lipid synthesis, and actually does the opposite, inhibit lipid synthesis. So FGF19 has this capacity of promoting glycogen synthesis and promoting protein synthesis in the liver, which fits very well. I'm going to go back once. Fits very well with what we have observed here. We observe decreased triglyceride lipid accumulation, increased protein synthesis. Fits very well with the effects that were described for FGF19 in liver. Up to here, we can only, speculate about the effect of FGF19 or the effect of bile acid through FGF19, in liver. But, in August 2017, actually a French group came up, with this data set showing that the effects of FGF19 are actually expanding beyond the liver and also affecting skeletal muscle. And they show compelling data that FGF19 regulate muscle mass and it has those effects independently of mitosis, independently of differentiation. So putting the whole story together, our data indicates that antimicrobials, what they do is they target in part these bile acid transformations by the gut microbiota. They enhance 7-alpha dehydroxylation, and thereby they determine a bile acid profile that is more potent agonist of bile acid sensors, that is FXR and TGR5. So activation of FXR in the intestine, where most of these bile acids are produced, that is where you expect to see a stronger, let's say, phenotype, and that is what we observe, inhibits inflammation or contributes to attenuate inflammation, but also, improves enteroprotective system like production of antimicrobial peptide and barrier function. And then these effects also expand systemically, in part by bile acids, traveling into systemic circulation and activating distally bile acid sensor expressed in adipose tissue, reducing inflammation locally there, but also targeting the liver. And in a more hypothetical situation, we also believe that FGF19 may be improving muscle accretion in skeletal muscle, protein accretion in skeletal muscle, sorry. So we continue on. So this is our, now state of the art, let's say, and we are taking this picture and expanding, looking at different angles here. One angle that we have been interested in, and we are more interested in now, is the potential effect of bile acids in regulating secretions. A lot of evidence or knowledge or published data is about the role that bile acids have in inducing diarrhea, but that happens when bile acids are at a pathophysiological level, when they are at very high concentrations for different reasons, and that has been very well documented. However, some newer data indicate that at physiological level, which is more on the left side of the panels that you have there, both DCA and CDCA, what they do is through FXR, they help in regulating the secretion, the intestinal secretion, particularly by regulating the secretion of chlorine. And they have been proposed to act as osmosensors that prevent excessive secretion of fluids in the intestine when a meal is being consumed. So we, are working on this, expanding on this, and I just remind you that we observe in our data set, reduce ion transport capacity, so short circuit current in one data set, and also we observe reduced intestinal permeability. So we decided to expand some of these. We are recently finished an experiment where we have taken pigs, again, very normal setting, nothing special about this, and we gave them oral garbage during 14 days of either water or DCA, the deoxycholic acid, which is a secondary bile acid, a potent activator of TGR5, and an FXR as well. So we use this, and at the end of this period, we use LPS challenge, which is a very well-established model of intestinal dysfunction. And we measured different things, and we found that DCA infusion actually prevented degradation of protein, tight junction proteins like Soluna occludens and Claudin 3, and that sort of preserved the localization of these proteins in the apical portion of the villi and prevented the internalization of the protein that is associated with the loss of intestinal permeability, or the increase in intestinal permeability, sorry. These changes were associated with attenuation in the concentration of circulating endotoxin, reduce inflammatory response. AP states for, in this case, is alkaline phosphatase, which is responsible for the deactivation of lipopolysaccharides. We observe an attenuation of that, and the concentration of bile acids in the ileum mucosa, as you can see there, there was a substantial increase of DCA, which is what we were infusing. So to summarize, I'm not sure how I'm doing with time. I did not keep track of that. I think I'm doing okay. So to summarize, just want to leave you with some of this message. We believe that bile acids are actually working at these signal transducing molecules in the immunometabolic interplay between gut microbiota and host. We believe that they are part of the mechanism that explains growth promotion by antimicrobials. That might also include metabolic regulation by FGF19 at the liver level, but probably also beyond liver, including skeletal muscle. We think that bile acids may play a critical role, particularly at weaning or stages where the gut development and maturation is compromised by signaling through TGR5 or FXR. But beyond antimicrobials, I think that we should, expand our ideas beyond these to also include situations pathologies that affect the gut, the liver, of the energy metabolism of food-producing animals. Certainly, any of them that affect intestinal dysbiosis, integrity, or also liver function. So with that, I'm pretty much done. So thank you very much once again for the invitation and your time and attention.